Hello students, welcome to Trunkie Academy. Today we'll study about the that was about we are studying about the cell and the under the cell unit of life we studied about the prokaryotic cell and eukaryotic cell. Okay, when we studied about the prokaryotic cell, we discussed about the different group of organisms that have prokaryotic cells. Unicellular organisms like bacteria, mycoplasma, PPLO, and cyanobacteria, blue-green algae have the prokaryotic cells. Okay. After that, when we study about the eukaryotic cells, we saw that there is a marked difference between the prokaryotic and eukaryotic cells. Eukaryotic cells are much more advanced in terms of the cell organelles and the nucleus and the packaging of the chromosomes, packaging of the DNA, and then we study about the plasma membrane. Now today we'll study about the cell wall endomembrane system, that is the membranes of the organelles and the endoplasmic reticulum. Okay, so first we'll start with the cell wall. Plant cells can be distinguished from animal cells by the presence of cell wall that I have told you many times before when I was discussing about the prokaryotic and eukaryotic cell. So that is one of the biggest difference between the plant cell and animal cells, where we find that cell wall is present only in plant cell, okay, not in animal cell. So now it was the first observed by Robert Hooke 1665 in the cork tissue of oak tree. He is one of the very famous scientists, and he was when observing the cork tissue of oak tree, and you know cork is a dead cells, dead tissue, and he found that there is some kind of compartmentalization in the tissue. So he, after some research and study, he came to know this is the cell wall. Cell wall is non-living rigid structure and forms an outer covering for the plasma membrane of fungi and the plants. So this is found in fungi and the plants. Okay, so in which tree Robert Hooke was it? Is a cork, and it was the oak tree. Okay, so cork tissue was also is an oak tree. So cell wall not only gives shape to the cell but also protects the cell from mechanical damage. Very important, and infection also. It's not only giving a characteristic particular type of shape to the plant cell, but also providing from the damage. It's providing because this cell wall is hard and rigid, okay, and also giving a protection from the infection. Cell wall also helps in cell to cell interaction and acts as barrier to undesirable molecules. Okay, there are some kind of communication between two adjacent cells, okay, where the cell walls are communicating. There is a kind of communication okay, link between two cell walls. Like for example, if this is the cell wall, okay, so this will continue. There will be another, there will be continuation, and then there is a cell which is there. We will have the communications. Okay, so there is a communication, and this is called the plasmodesmata, the connecting link. Okay, so algae have cell wall made of cellulose, galactans, mannans, and minerals like calcium carbonate. There is some differences between in the chemical composition of cell wall in different group of plants and different group of organisms. For example, when we observe the cell wall of the bacteria, it is different from the cell wall of green plants. Okay, algae have cell walls made of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectin, and proteins. So this is the marked difference. You can say a clear-cut difference between the cell wall of algae and the higher plants, okay, or in the higher green plants. So cellulose and the hemicellulose. Cellulose is common, but there is galactans and mannans and mineral like calcium carbonate is present in the cell wall of algae. Okay, this is very important for the NEET examination. Okay, so cellulose, hemicellulose, pectin, and proteins are present. In case of this, the algae, galactans and calcium carbonate, mannans are present. Mannan is also kind of carbohydrate. Okay. Okay. The cell wall of young plant cell primarily wall is capable for growth. Okay, in the young cell, which is newly divided cells. Okay, young plant cell, the primary wall, or the primary and secondary wall is there. Primary wall gradually diminishes as the cell matures, and the secondary wall is formed on inner side of the cell towards the membrane. Okay, so what happens? There is a primary cell wall formed in a newly divided cells that matures, diminishes, and the secondary wall is formed, okay, on the inner side of the membrane. The secondary wall is formed by the addition of new cell wall materials like lignin, suberin, pectin, and cutin into cellulose interfibrillar species. Okay, so when this primary wall diminishes and the secondary wall is being formed, there is addition of some hardening substances. These lignin, suberin, pectin, and cutin are the substances that confers the hardness of the cell wall. They are responsible for providing the rigidity in the cell wall. Okay, these substances are cementing material and acts as cementing material and provides the hardness. Okay, in the interfibrillary. Now in xylem, the secondary wall exhibit unthickened thin areas called pits to support intercellular transport. So there are some kind of like connection, you can say gap or thin unthickened areas are there for transport. Okay, intercellular transport, communication between the two adjacent cell. Okay, so you can see cell, middle lamella, secondary wall, primary wall. So as I told you earlier, primary wall is found that is diminishing, and secondary wall is found towards the inner side, towards the plasma membrane side. So there is a middle lamella between the two cell wall. You can see there is a middle lamella and the primary wall, and then the secondary wall. Okay, what are unthickened areas in xylem? Pits. Okay, there is a space. Okay, the middle lamella is mainly composed of calcium pectate. This is an important point. Important for exam. Middle lamella is mainly composed of calcium pectate. It's a kind of chemical compound which also provides rigidity, and this is present in the middle lamella between the two cell wall of the two different cells. Okay, so middle lamella is the first formed layer of cell wall and develops from the cell plate during cell division. Important point to remember. Okay, develops from the cell plate during cell division. Middle lamella develops from this during cell division. And in a mature plant cell, the middle lamella is the outermost layer. Yes, obviously, because when there is two cells, adjacent cells, there are middle lamella, then the primary wall, primary cell wall, and then the secondary cell wall. Okay, so first is the outermost is the middle lamella. So pits means porous primary cell wall separates adjacent tracheids and the vessel elements. That is the pits. Okay, and sclerenchyma cells are there that you will now. Okay, tracheids, vessel elements, and complete perforations. You can see. Now you can this in this picture you can see, understand. Okay, you can see the middle lamella, the primary wall, then the lumen, the secondary wall. Okay, and you can see in this way. So if you cut the plant like this, and transverse section. Okay, and if you cut like this longitudinal, so then also you can observe the middle lamella, then primary wall, then secondary wall, and there is the S1, S2, and S3. That means the different the parts of the cell. The middle lamella is composed of calcium pectate, that I told. The cell wall and the middle lamella may be transversed by plasmodesmata, connecting beam, that I showed you in the diagram. Connection between the two adjacent cells, okay, through the cell wall, the plasmodesmata connects the cytoplasm of neighboring cells. Connection, endomembrane. Okay, this is here. I will just show you the diagram. Like I told you, there will be communication between the two cells. This is very small. I can say you the is very small and connection, and this is connecting the another cell. So this connection, where you have the another cell, here you have another cell, and this will have a connection between like this. So connection between the two cytoplasm. Okay, here also the two cells are there. Okay, and this is the connection. It's connecting the two cytoplasm. That is the plasmodesmata. Endomembrane system. Many of the membrane organisms are considered together as an endomembrane system because their functions are coordinated. Now, what do you mean by this endomembrane system? Endomembrane system means within the cytoplasm there is kind of because this is the functions, and within the cell is also acting as a one living entity, and it has got some important functions and role to play for the tissue in together. Okay, so what happens? There are endomembrane systems. That means there are some kind of organelles and some kind of vesicles are formed that within the having membrane and within the cytoplasm. Endomembrane includes the Golgi complex, lysosomes, vacuoles, endoplasmic reticulum, etc. So these are the endomembrane. Within the cytoplasm, there are some important organelles. They are having with the membrane, and they play important role in the function of the cells and then the functions of the individual body. Okay, so Golgi complex, lysosomes, vacuoles, and endoplasmic, they have got the membrane, and under they are forming the endomembrane system. Now, mitochondria, chloroplast, and peroxisomes are not included in endomembrane system because their functions are not coordinated. Oh, now another important point I forgot to tell you that Golgi complex, lysosomes, endoplasmic, they are all are coordinated. For example, if endoplasmic reticulum is sending some substances after processing, the Golgi complex will further it will process and changes and modify. Then set it is the lysosomes. Then lysosome goes to the vacuole. So all these are coordinated, and then the functions are interrelated. So once the function of the endoplasmic reticulum it passes on to Golgi complex, then it passes on the lysosomes. Then the product of the lysosomes will goes to the live vacuole. So in this way, these are coordinated, they are interrelated, so are endomembrane system. But in case of this mitochondria, chloroplast, and peroxisome are not endomembrane system because their functions are not coordinated with endomembrane system. Okay, so they are the same. Now, endoplasmic reticulum, one of the most important part of the cell, or you can say cell organelle. You can see this. The picture is very clear. Nuclear envelope, the part which has been shown, okay, in the purple, that is the nucleus, okay, and the nuclear envelope you can see. Now this endoplasmic reticulum is attached on the nucleus, okay, and there are some granules like structure, small, spherical, that is the ribosomes, and hence these endoplasmic reticulums are called rough endoplasmic reticulum, as this looks rough because the presence of the ribosomes on the surface of the endoplasmic reticulum. And then you can see the smooth endoplasmic reticulum where you do not find any ribosomes on its surface. Okay, so endoplasmic reticulum are of two types: rough endoplasmic reticulum and smooth endoplasmic reticulum. K.R. Porter, the scientist in 1953, first coined the term endoplasmic reticulum to describe the thread-like structure in the cell, thread-like small structure in the thread, and he coined this term in 1953. Okay. The studies of eukaryotic cells reveal the presence of reticulum of tiny tubular structure scattered in the cytoplasm that is called endoplasmic reticulum. Endoplasmic means the tubular structure is stack-like and flattened, okay, and they are present in the near the nucleus or attached with the nucleus. Endoplasmic reticulum divides the intracellular space into two distinct compartments. Within the intracellular space, the cytoplasm, they are dividing into two distinct compartments. That is the luminal inside the ER, okay, and the extraluminal cytoplasm. So the compartment that within that they within the lumen of the endoplasmic reticulum, that is one chamber or one compartment, and then outside the endoplasmic reticulum, that is extraluminal, okay, that outside the ER, that is the cytoplasm. So that it divides into the two. In this way, it is considered as the two compartments. So you can see the smooth endoplasmic reticulum without any ribosomes attached on it. Then the cisternae. Cisternae are this flattened sac, like that is the cisternae of the endoplasmic reticulum, and with the presence of the these ribosomes, these are considered as called the rough endoplasmic reticulum. You can see the lumen of the cisternal space, lumen, stack-like, okay, lumen. There is space in the cisternae, and the rough endoplasmic is here, and the nucleus is this side, and this is attached with the nucleus, and the ribosomes are attached. So you choose. This picture is to mainly show the cisternal space, the lumen of this lumen. This is the lumen, and this is called the one compartment, and outside this is the another compartment, that is the cytoplasm. Okay, so endoplasmic reticulum, rough endoplasmic and smooth endoplasmic, that I have already told you. Now endoplasmic reticulum, you can see the cisternal space, nuclear envelope since it is attached, and there is the nuclear pore, okay, yellow, green, you can see. So that is the nuclear pore. So small pores are present on the nuclear envelope, or you can say nuclear membrane. Okay. Rough endoplasmic reticulum, they are smooth. They are with the presence of these ribosomes, so they are rough. RE are strictly observed in the cells actively involved in protein synthesis and secretion. These are involved mainly. Now, this function is also important point. They are involved in. This is important for exam again. Okay. They are frequently involved or absorbed in the protein synthesis and secretion. Okay, rough ER. And are extensive and continuous with the outer membrane of the nucleus. So they are, as you have seen in that picture also, the diagram, they are continuous with the outer membrane of the nucleus. Okay, they are continuous. They are okay. So this is the they are areas extensive and continuous with the outer membrane of the nucleus. Continue. Okay, so outer membrane of the nucleus, and then forming the endoplasmic reticulum. Now, rough endoplasmic reticulum with the ribosomes, and smooth endoplasm in the absence of ribosome. Okay, and they are involved in synthesis of lipids. So remember this. In case of the rough ER, they are involved in the synthesis of protein and secretion. Here, this SER, smooth endoplasmic reticulum, is involved in the synthesis of lipids. In animal cell, lipid like steroidal hormones are synthesized by SER. Okay. Now, smooth endoplasmic reticulum, they have got membranes. Okay, and they do not have ribosomes attached on it. So, in MCQ, who first observed the cell wall? The Robert Hooke. The Robert Hooke in 1665. Okay, Robert Hooke, not Singer and Nicholson. Singer and Nicholson were the scientists to give the fluid mosaic model. Okay, Robert Brown is another scientist who observed the Brownian motion, and he observed about the studied about the pollen grain. Okay, so it is the Robert Hooke in 1665. Outermost layer of the cell wall, it is the middle lamella, and primary cell center, no, it's the middle lamella. Now, plant cells are connected by channels through their walls called plasmodesmata. Okay, so I told you. Okay, an extensive network of tiny tubular structures scattered in the cytoplasm continues to the outer membrane of the nucleus is the endoplasmic reticulum. Right? Okay, so wherever you observe the diagram or the picture of the cell, you'll find that endoplasmic reticulum is attached near the nucleus. Okay, so it is continuous with the outer membrane of the nucleus. The following cell are not included under the endomembrane system. This is the mitochondria. Okay, mitochondria is not in coordination with all lysosome, Golgi complex, and ER. So now we'll study about the Golgi apparatus or Golgi bodies or lysosomes and then vacuoles. Okay. So Golgi apparatus, it has got Camillo Golgi. He is the scientist. He discovered this and observed densely reticular structure near the nucleus. Okay. We came to know that endoplasmic reticulum is found attached to the nucleus, and this Golgi apparatus is also found not attached, but near the nucleus. It was first observed by Camillo Golgi in 1898, and he observed that it was a densely or highly stained structure, reticular structure near the nucleus. Okay, while staining with some dyes and stains, he found that some densely reticular structure are there. So he is the gentleman scientist who discovered this, Camillo Golgi. Okay, Golgi apparatus consists of many flat discs. Okay, flat disc sac or cisternae of 0.5 micrometer to 1 micrometer in diameter. Okay, so you can see in the picture, these are stacked parallel to each other, and varied number of cisternae are present in Golgi complex. You can see this is the cut portion. So you can see the lumen. Okay, so this is the lumen part. This is the lumen, the cut portion, and so slowly these cisternae are forming a vesicle at the terminal, at the end, and then ultimately these vesicles are getting detached from the cisternae and forming the vacuoles and going to the other part of the cytoplasm. Okay, and there is vacuole also, you can see here. Okay, so this is the vacuole. Okay. So cisternae. Okay, this stake-like. Okay, a flat there is a lumen, and there is stake-like, and there is one by one that forms the stake-like cisternae. Now, Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and the convex trans or maturing phase. There are two phases. One is forming and one is the maturing. So the two phases, the two sides are there. So okay, so you can see the cis receiving the sides of receiving side of the Golgi apparatus, receiving side from the materials are received for the processing. So receiving side, and another side is the trans or maturing phase. So you can see the two sides are there. Okay, or two part you can say of the Golgi apparatus, the cis and trans. Cis means the forming phase, which is receiving the side, receiving end, and one is the maturing, from where the vesicles are getting detached. Once formed and processed, they are going into the other part of the cytoplasm. Okay. The Golgi complex function is packaging materials to be delivered to intracellular targets within the cells. Okay, the vesicles which are being detached from the cisternae. Okay, they are going to some different parts of the cytoplasm as their targets. Okay, or secreted outside the cell. Sometimes they are going to the plasma membrane and then they are liberating their content to, and then it goes to outside the plasma membrane or secreted outside the cells. Okay, so this way their main function is to packaging the material that has been come from the endoplasmic reticulum, that is modified, processed and packaged by Golgi complex, and then it is delivered and then it is transported in the form of the vesicles, whether it is required in the within the cytoplasm, within the cell, or secreted outside the cell. Okay, materials to be packaged in the form of vesicles from ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This I told you earlier that when the ER has got some kind of products, they are sending it to the Golgi apparatus, and it comes to the cis phase, forming phase, and then it processed, packaged, and then it moves to the trans side, and from there it forms a vesicle, and that vesicle will detach, cut off from the cisternae, and then move to the cytoplasm or go outside to the liberates content outside the cell. Okay, so that is how it works. This explains why Golgi apparatus remains in close association with the endoplasmic reticulum. That is the reason it's present near the nucleus or endoplasmic reticulum, because they need to receive the materials from the endoplasmic reticulum. Proteins are synthesized by ribosomes on endoplasmic reticulum. Okay, ribosomes are the factories of protein. So proteins are further classified, further modified in the cisternae of Golgi apparatus before they are released from its trans phase. Okay, so what happens? We know that ribosomes are the cell organelle where the synthesis of protein takes place. Okay, and proteins are synthesized by ribosomes on the endoplasmic reticulum. Then these are passed to the Golgi apparatus, where these proteins are modified, okay, in the cisternae, and then they are released from the trans phase of the Golgi apparatus. In plants, Golgi apparatus involved in the synthesis of cell wall materials and also plays an important role in the formation of cell plate during cell division. So in case of plant cells, it's also involved in the formation of cell wall materials for the cell walls. Okay, but not in animal cells, since in animals do not have cell wall. So they are basically involved in the packaging and processing of the substance of protein that will be received from the endoplasmic reticulum and ribosomes. Okay, Golgi apparatus is important site of formation of glycoproteins. Yes, glycosylation. For example, the protein is synthesized in the ribosomes. Now that has moved to the Golgi apparatus. Now they are going to glycosylation, going for the glycosylation or attachment of some kind of carbohydrate to the protein, or some kind of modification and packaging. All these things takes place in the Golgi apparatus. Glycoproteins are also formed in when the proteins are getting attached or being associated with some kind of carbohydrates. Okay, so that becomes a glycoprotein, and that happens at the Golgi apparatus. Okay, or glycolipids. Okay, so when the lipids are formed and they are there, and the glycosylation of lipid takes place, then into the glycolipids. Okay, it forms the glycolipids. Okay, now lysosomes. Lysosomes are also important. Lysosomes are rich in hydrolytic enzyme, hydrolysis, powerful enzymes that will completely destroy this or burn the cell organelle. Okay, or the whole cell. Okay, powerful hydrolytic. Okay, so digesting enzymes. You can say hydrolysis, digesting, powerful digesting enzyme or oxygen. Lysosomes are membrane-bound vesicular structure found by the process of packaging of the Golgi. So now, when from the transform of the Golgi apparatus, when the vesicles are detaching away, getting forming the vesicles and moving the small vesicles where they form, and then they become the lysosomes. Okay, the isolated lysosomal vesicles are found to be very rich in hydrolytic enzymes. Okay, so this is what it means. Lysosomes are membrane-bound vesicular structure formed by the process of packaging in the Golgi apparatus. They are formed by the packaging in the Golgi apparatus, and then are released and they are moved in the cytoplasm. Okay, So isolated lysosome vesicles are found to be very rich in, as I told you earlier, they are filled up with hydrolytic enzymes, powerful oxidizing and hydrolytic enzymes, hydrolysis. Okay, hydrolysis is capable of digesting carbohydrate, protein, lipids, and nucleic acids. That is the reason they are able to completely destroy or digest the cell when they burst, when they break, and liberating out their content. They are completely digesting the cell. Okay, and are also called suicidal bag of the cell. The example of hydrolytic enzymes found in lysosomes are lipases, proteases, carbohydrases, and nucleases. These are the enzymes: lipases, proteases, and carbohydrates, hydrases, and nucleases. Okay, now anatomy of lysosomes. Okay, you can see the lipid bilayer. Okay, plasma membrane has got lipid bilayer, and this is the this has been this plasma membrane. This part has been shown in a magnified view here, magnified view. Okay, so we can see the plasma membrane. Okay, and then we can see the hydrolytic enzymes are present. Okay, and glycosylated membrane transport protein, okay, are present on this plasma membrane. Okay, so this is how it looks like. Now lysosome is structure: cell wall membrane, enzyme complexes. The hydrolytic enzymes are optimally active at acidic pH. Okay, optimally means they are in to optimally, okay, exactly, clearly becoming active, okay, I mean at its full capacity, or fully capable becomes at the acidic pH. Okay, so at alkaline pH also they may be active to some extent, but they are best active at acidic pH. So what is now we question comes, what is autolysis? Because since they are called suicidal bags of the cell, because when they burst, when they're liberating their content, whole cell is digested. Okay, so under starvation conditions, lysosomes digest cellular contents by releasing hydrolyzing enzymes and cause death of the cells. This is called autolysis, and this is important thing. What is autolysis? The death, causing the death of the cell by liberating the enzymes that is present in the lysosomes. Okay. Vacuoles. Now another one I told you, vacuoles are membrane-bound space found in cytoplasm and most common to plants. Okay, these are mainly found even in animals also. It is found, but it is mainly found and most it is common to plants. And these are one single membrane-bound space that may be filled with water or gas. Okay, so here you can say tonoplast. That the membrane that it's called the tonoplast. Okay, cell sap or central vacuole. You can see that central vacuole is there. Cell sap is present. Okay, and this is the vacuole. Okay, water with dissolved minerals. It's the cell sap, water with dissolved minerals. So what is tonoplast? The vacuole is bound by a single membrane, that is the tonoplast. So this is important exam. A vacuole sap is composed of water, then the excretions, excretory product, metabolic byproducts, and waste materials. Okay, so these are the vacuole sap is composed of. Okay, water, excretions, or excretory products, metabolic byproducts, waste products. Okay, in some plant cells, vacuolar sap also contains some pigments like anthocyanin, which impart color to the plant part. Like purple color is imparted by the anthocyanin. This is the anthocyanin. Okay, this is a structure, chemical structure, the anthocyanin, which impart color. Now, normally what happens? The pigments that is present in the carotenoid or the chlorophyll, okay, or chromophyle, okay. But this anthocyanin provides the purple. In plant cell, vacuoles can occupy up to 90% of the volume of the cell and play important role in osmoregulation. Okay, so these vacuoles can occupy about 90% of the total volume of cell, and play important role in osmoregulation, maintaining the ion concentration. Okay, osmoregulation. In plants, the tonoplast facilitates the transport of number of ions and other materials against concentration gradient into the vacuole. So this tonoplast playing an important role and facilitates the transport of number of ions and other materials against concentration. So while transporting against the concentration gradient, these tonoplasts are playing important role and facilitating the transport of a number of ions. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. So that is the reason the ions Okay, the concentration of iron is very high in the vacuole than the cytoplasm, because the chloroplast is allowing the movement. What is the important role of vacuole? It is the osmoregulation. Okay, like 90% of the cell is occupied, volume is occupied by the vacuoles, because for osmoregulation. Okay, to maintain the ionic balance. So vacuoles are of three types: gas vacuoles that have only gas, contractile vacuoles, and food vacuoles. Okay. Now in amoeba, contractile vacuole is important for excretion. Whatever the excreta, metabolic waste is formed or produced in the cell, okay, in the amoeba, that is collected and goes into the contractile vacuole, and that again that burst and release its content, okay, in the environment. Okay, so the contractile vacuole, okay, pseudopodia, the engulfing the food. Okay, the food vacuole is there. Now after the utilizing the food, okay, metabolic waste is formed, and then it goes to the contractile vacuole. In many cells, as in protist, food vacuoles are formed by engulfing the food particles. Okay, like in amoeba also, we form the food vacuoles, or in the paramecium. Okay, so after engulfing, they are forming the food vacuoles. Like we have seen here, food vacuole. Food vacuole is this. Okay, this forms the food vacuole. This one, food vacuole. After engulfing, the pseudopodia are formed, and then they are going around the food, okay, and slowly they are merging and then forming, then engulfing and forming the food vacuole. Okay, so oral groove sweeps food into food vacuoles. So in case of this paramecium, they have got oral grooves. Okay, paramecium is an example of the protist. Okay, like amoeba, this is also protist, and they have got some oral groove, and they are just sweeping the food into the food vacuole. Okay, so you can see the food vacuole is there. Food vacuole, this one, food vacuole, right? Correct. So this is also the food vacuole. Okay, so this way now protist engulfing the food particles here and forming the food vacuole. Okay, so an amoeba feeds small organisms such as bacteria. Okay, and you can see then it is the pseudopodia are closed and then forming the food vacuole. Okay, so digestive enzymes break down the food and the nutrients diffuse it into the cytoplasm. So digestive enzymes that is present in the cell, that will breaks down the food, and then nutrients will diffuse into the into the cytoplasm. Okay, this is how the food vacuole is formed. Now we're coming to some MCQs. This cell organism helps in digestion process in plants. Centrosome, lysosome, lisosomes, and plasma membrane. Digestion process means not like they're eating food and they need to eat. Digestion process means if the cell is damaged and destroyed, it's no longer functional, and if the cell is due to some reason its function is over, it's no longer required, okay, or damaged, then there will be bursting of the lysosomes, and then there will be release of the or liberation of the content that is present in the lysosomes, the hydrolyzing enzymes, the powerful hydrolyzing enzyme that will oxidize, that will digest the whole cell, and that will clear up. Okay, in this way it will destroy it. So that is called the reason suicidal bag of the cells, or autolysis. Okay, when it's not needed, then autolysis occurs. So lysosomes is the food digestion. Food digestion means we need to say means there is a lipid, there is a nucleic acids, there is the carbohydrate. Okay, so in that way, if you think it's a food digestion process, but it is required to destroy or clear up or digest the cell. Okay, now hydrolytic enzymes are abundant in lysosomes. An old plant cell is characterized by absence of vacuole or presence of two nuclei, or absence of nucleus, or presence of large vacuoles. It is the presence of large vacuoles, because old plant cell will, when it matures, a large vacuole will be present, because it need to there has to be some osmoregulations. Okay, so large vacuoles are present in the matured plant cell, and the plant cell which has been just divided and newly formed, there will be no large vacuoles. Okay, so there may be absence of vacuole, and slowly there will be a small vacuole, and as it matures, it become matured, then it become the there will have the large vacuole. Okay, now the vacuoles contains phycocyanin, anthocyanin, xanthophyll, and phycoerythrin. Okay, so this is how this the type of the vacuoles contain it is anthocyanin. Okay, it is anthocyanin that gives the imparts color to the certain plant part. Even in the some petals of the flower, there is anthocyanin imparts color like purple and violet color of the petals. Okay, so anthocyanin in dissolved form. Now, densely stained reticular structure near the nucleus is the Golgi complex, Golgi apparatus or Golgi complex. Okay. Now, this was all about the endomembrane system. Okay, Golgi apparatus, endoplasmic reticulum, Golgi apparatus or Golgi complex, lysosomes, and vacuoles. So they are coordinated in their functions. So they have got some important role. When we talk about the endomembrane system and when we study about the endoplasmic reticulum, there are two types: smooth and rough. Production of the synthesis of the protein takes place in the ribosomes. Okay, and some substances are produced and synthesized and secreted in the endoplasmic reticulum, and these products when reaches the Golgi apparatus, okay, processed and packaged and then released into the cytoplasm. And lysosomes, which are comes from the after processing from the Golgi apparatus, they have got some powerful enzymes or some hydrolytic enzymes, okay, and that can digest carbohydrate, lipid, nucleic acids, etc. And vacuoles are a large, okay, and that may contain liquid or gas, and they've got important role to play in plant cells. Okay, vacuoles is mostly found in the plant cells. Okay, so it's main characteristic feature of the plant cell. So these were all about the endomembrane system. In the next class tomorrow, we will study about the next topic. So now tomorrow we will again meet at the same time. Now till then, goodbye and take care.